Hello everyone. Today we will talk about oncogenes and their mechanism. Now oncogenes we will first discuss about its discovery. Uh, Oncogenes can be traced back to 1970s where researchers began to identify the genes that were associated with cancer development. Now the term oncogene was coined by Dr. Peter Vogt in 1976. It was to describe a gene that could transform normal cells into cancerous cells. Okay. Now, the early discoveries in the field of oncogenes was the identification of Ross sarcoma virus. Ross sarcoma virus, that is RSV, which was identified as a cancer causing agent. It was found to contain a gene called SRC. RSV, that is Ross sarcoma virus, was found to contain a gene known as SRC, which was responsible for transforming the normal chicken cells into the cancerous cells. Now, this was later identified as an oncogene, right? Now, further research led to the discovery of other oncogenes, including RAS gene. Different genes are discovered, right? MIG gene, RAS gene, ERB1 gene, various were discovered. We'll talk in a brief about them. First is MIG gene, right? MIG gene plays a vital role in growth of cells and division of the cells, right? And they code for transcription factors which regulate the cell proliferation and growth and differentiation, right? Transcription factors which regulate cell proliferation, growth and differentiation. Now, MIC family, it is a family of genes having three members, CMYC, NMYC and LMYC. We'll talk about it later. Now, ERBB gene or EGFR, which is epidermal growth factor receptor gene. Epidermal growth factor receptor gene encodes for cell surface receptors that are involved in the cell growth, differentiation, and survival. Right. As- Now, the mutation or overexpression of this gene can implicate in the development of various types of cancer like breast cancer, lung cancer, colorectal cancer, colon cancer, etc. And the discovery of the role of ERB genes in cancer has led to the development of targeted therapies, right? Discovery of ERBB gene has led to the development of targeted therapies that specifically block these receptors such as a different ERB for different ERBB positive breast cancer, right? RAS gene. RAS gene codes for proteins involving in the cell signaling pathways. Specifically, they are small GTPs that act as molecular switches to control a variety of cellular processes, including cell growth, differentiation, and survival. Now, mutations in this gene can lead to the constant activation of this signaling pathways, promoting uncontrolled cell growth and proliferation and cause cancers like pancreatic cancer. Right. Now, we will talk about a little bit more about RAS gene. We already know that RAS gene encodes a protein that is involved in we can remember that this is important about RAS gene, right? RAS gene encodes a protein that is involved in regulating cell growth and division. Now, mutation in this can lead to uncontrolled cell growth of the cells, which will ultimately lead to the development of cancer only, right? And this RAS gene was found to be mutated in a significant percentage of human cancer, right? So RAS gene is important. Now, now we will talk about proto-oncogenes. They are nothing but normal genes, right? Which are present in our body performing normal functions. They encode for proteins, various proteins in our body, For example, growth factors or growth factor receptors. Second can be transcription factors and signal transduction factors. There are many more. These are some examples. Right. These are proto-oncogenes. Right. Now, what happens to them that they can get converted into abnormal genes that are known as oncogenes? Right. We will talk about it. So, what happens to them that they somehow get converted into oncogenes? What happens to them? Right. That they become oncogenes? Why and how? This will be discussed in the mechanism of proteogene activation, which we talk about in detail. Now, first I want to discuss that proto-oncogene becoming an oncogene it can either by three methods. These are the three methods of activation of oncogenes. Which we will discuss in the next. The mechanism of oncogene activation has three methods. First is mutation, second is gene amplification, and third is chromosomal arrangement. Third is chromosomal rearrangement. Mutation, either by mutation in this normal cells or proto-oncogenes, now we will discuss about mechanism of protogene and oncogene. Now we will talk about mechanism of oncogene activation. Mechanism of oncogene activation, that is there are three methods. First is mutation, second one is gene amplification, and third is chromosomal rearrangement. These are the three. Now first we will talk about mutation. Mutation, in point mutation, what happens at the DNA level? As we know that point mutation happens at the DNA level, and where there is a mismatching of base pairs happen, right, which occur during replication, right. When replication is undergoing, there is some mismatching or there is some error in the base pairs, right, and this base remains in the strand when the DNA repair is unsuccessful, right? It fails. That DNA repair fails, due to which the DNA strands still have this mismatching of that base pair, or the base. In this base, you can see that we can see that here is AGG, this many codons, 5 codons are here, and their complementary codons are also here. And what happens is that a single base pair gets mismatched. Here C, that is cytosine, should have been paired with guanosine, right? But what happens is that here comes A, which is the adenosine, and it gets impaired, right? And due to which, when transcription happens, the mRNA has the same complementary of that, and which is UGA. U comes here, and C is complementary is G, and T is complementary is UGA is a stop codon, due to which it gets halted. And when an protein is translated from this type of mRNA, that protein is altered. That altered form of protein, which is not functional, right? And due to which the oncogenes are formed, right? Now, regulatory mutation. Regulatory mutation is a type of mutation when enhancer sequences, right? When enhancer sequences of the gene of the DNA are mutated, and the regulation of the gene is compromised. Thereby, the normal genes overexpress it in that case, right? Overexpresses that in that case, right? And example, always remember that example of this point mutation was RAS gene. That's why I told you that RAS gene is very important. RAS gene. RAS gene is an example of point mutation. Remember it. Now we will talk about gene amplification will come, right? Now we will talk about gene amplification. Gene amplification is basically the normal expression gets amplified, right? Amplification of the normal gene expression. Now we will talk about gene amplification. Gene amplification is simply but the amplification of the normal gene expression, right? Normal protein gets overexpressed, which ultimately leads to the formation of oncogene. Now, chromosomal rearrangement. Chromosomal rearrangement can happen in the regulatory sequence or the protein coding sequence, right? When it happens in the regulatory sequence, the normal protein gets overexpressed, and when it happens in the protein coding sequences, it causes the fusion of two genes, right? Now we will talk about this. Some examples of this chromosomal rearrangement, right? For example, growth factors in SIS gene. SIS gene, it encodes for a beta chain of PDGF, right? Right. It encodes for the beta chain of the PDGF. Now we will talk about some examples of the proto-oncogenes. For example, first is here. Now we will talk about some examples of the proto-oncogenes. First is the growth factors in SIS gene, which codes for the beta chain of PDGF. PDGF is the platelet derivative of the growth factor. Now we'll talk about some examples of proto-oncogenes. Proto-oncogenes, first growth factors, SIS gene, which encodes for the beta chain of PDGF. PDGF, that is a platelet derived growth factor, and mutation in this SIS gene can lead to the formation of SIS oncogene, which ultimately performs a neoplastic transformation of fibroblast, right? The second is nuclear proteins. Nuclear protein, that is CMYC, NMYC, and MYB. Now the translocation of the CMYC gene with IgA gene. IgA gene is a very important gene in which leads to Burkitt lymphoma, right? This will be studied in further classes. Now AB1 gene. AB1 gene, this aids in cell division, cell adhesion, stress response, but when it undergoes chromosomal rearrangement with the BER gene, right? what happens is that it undergoes a chromosomal rearrangement of BCR gene, due to which oncogene gets formed, which causes chronic myeloid leukemia. Right? This is the gene. Now there are many more examples, like now there are some more examples, like RAF gene. Now RAF gene, it is a part of ERK pathway, right? It, and the mutation in this valine, which is the amino acid at 600 position, causes the incorporation of the glutamine instead, right? Now, which renders the kinase domain to become active permanently, due to which the ERK pathway remains continuously on, which will lead to the ultimately lead to the continuous cell division, which will ultimately lead to the cancer, right? So what happens in RAS gene, right? It is a part of the ERK pathway, due to which we can say that mutation in this gene can how it happens is that valine, instead of valine, it codes for glutamine instead, right? Glutamine, due to which what happens is that the kinase domain remains active permanently. Kinase domain becomes active permanent. It becomes active permanently, right? Due to which the ERK pathway, which that RF gene is involved in, also becomes continuously in on position, right? It becomes continuously in on position, due to which what happens is that the cell division occurs continuously. Now when the cell division occurs continuously, what happens when cell division what happens when cell division occurs continuously? The cell will divide continuously, and there will be a mass of cells formed. With what does it mean, that mass of cells? Cancer only, right? Which will never die. It will continuously divide. And we can discuss more about these genes in further videos. Thank you very much.